So we'll move right on and hear from Niamh Mullins, who has been leading some of the bipolar disorder working group analyses, and she's based at the Icahn School of Medicine at Mount Sinai. Niamh.
Thanks, Jordan, and thanks for the invitation to present. I'll be sharing the results of the PGC3 bipolar disorder GWAS on behalf of the Bipolar Disorder Working Group. Firstly, this is where we're at in terms of published results in bipolar disorder. The PGC2 paper came out last year in Nature Genetics. It had a sample size of 20,000 cases and identified 30 genome-wide significant loci, implicating many genes involved in neurotransmission. SNP heritability was 23%, and polygenic scores explained about 4% of variance on the liability scale. And a key part of the paper was in highlighting the differences between type 1 and type 2 bipolar disorder in terms of their SNP heritability and their polygenic signal, with type 1 being the more heritable of the subtypes and showing a closer genetic relationship with schizophrenia, compared with type 2, which is more genetically similar to depression. So in PGC3, we've doubled the sample size to about 42,000 cases. The meta-analysis consists of 57 cohorts, still they're all of European ancestry. And amongst these cases, we have several subtypes of bipolar disorder. Most of them have been ascertained using structured psychiatric interviews, except for the external studies. These are several biobanks that we're collaborating with, which typically ascertain using ICD codes or self-report measures. So within PGC, all of the data have been processed using Ricopili as standard, and we've updated the imputation to the HRC. We have 64 genome-wide significant loci, 34 of which are novel for bipolar disorder. These are the ones shown in yellow. SNP heritability is 18% on the liability scale. And taking a look at the novel loci, these are the novel loci labeled by the nearest gene to the top lead SNP. So the MHC is now genome-wide significant. We also see two more of the CACNA calcium channels coming up. The locus involving furin, which is well known for its role in schizophrenia. And of the novel loci, many of them have previously been associated with schizophrenia at genome-wide significance. We see the first evidence of genetic overlap in terms of individual loci with major depression. And we also have two overlapping with childhood onset psychiatric disorders. So we have integrated eQTL data, and we're also doing statistical fine mapping of the GWAS results, which should get us closer towards the causal genes within these loci. This is how the signal looks in the extended MHC in bipolar disorder compared with schizophrenia. So we're looking at one signal MHC in bipolar disorder. We don't see this second peak around the C4 genes that's seen in schizophrenia. And through collaboration, we have been able to impute the C4 structural variants and C4 expression levels using our genotype data and a new C4 reference panel that's available. While initially there's a weak association between predicted C4 expression and bipolar disorder, after conditioning on the top lead SNP, this completely disappears, and there's no other peak here coming up around C4 as there is in schizophrenia. So while the MHC is implicated, we have no evidence that it's driven by C4. We use MAGMA to look at signal enrichment in our GWAS results. There was significant enrichment in 10 pathways, all of which are involved in neurotransmission, neurogenesis, or calcium signaling. Looking at gene expression, we saw significant enrichment in genes expressed in all of the brain tissue types from GTEx and no other tissue types, with the strongest enrichment in the prefrontal cortex of the brain. And in terms of the specific cell types involved in bipolar disorder, here we have looked at this in 5 human and mouse brain data sets, which have single cell data available. And we're looking at the top 10% of genes most specifically expressed in a particular cell type compared with all other cell types in the data set. So the biggest enrichment we see is in neurons in the frontal cortex, but also neurons in several other brain regions. We see enrichment in medium spiny neurons and pyramidal neurons. These are similar to previous reports in schizophrenia, and we also found novel evidence of enrichment in interneurons in the prefrontal cortex and the hippocampus. We looked at enrichment in the gene targets of about 150 drug classes, and there was significant enrichment in the gene targets of 4 drug classes at the very broadest level. The top class being the psycholeptics, which includes the subclass antipsychotics. So it's reassuring that we reliably detect the known bipolar disorder treatments. But we also see enrichment in the targets of several other classes, such as calcium channel blockers, which are used for treating hypertension, also drugs for treating functional GI disorders, which include anticholinergic drugs, and the gene targets of anti-epileptics. So these point towards new therapeutic leads and potential opportunities for repositioning some classes of drugs as potential bipolar disorder treatments. We've integrated eQTL data in several ways in order to prioritize biologically relevant genes from our GWAS. Firstly, we performed a transcriptome-wide association study using the TWAS fusion method and data from PsycEncode, which is the largest brain eQTL reference panel available. We then used Focus, which is a Bayesian tool for fine mapping, to fine map any regions where there was TWAS significant genes. And we also ran SMR using data from PsycEncode and the eQTL gen blood panel. And SMR additionally implements the Heidi test, which suggests that SNPs are causal for bipolar disorder via gene expression. So we're integrating the results from several methods here. Firstly, using TWAS fusion, there was 77 TWAS significant genes in 40 regions. So here, risk alleles for bipolar disorder affect the expression of these genes in the brain. And the region is defined using the correlation matrix of predicted expression from TWAS fusion. So this motivated us to fine map these 40 regions using Focus. And Focus fine mapped 30 of these regions, prioritizing only one gene that had a high probability of being the causal gene within that region. And as a further sensitivity analysis of those 30 genes, these are the ones which are also SMR significant for association with bipolar disorder, and they pass the Heidi test, meaning that we've been quite stringent with the criteria here. So these are 13 high confidence genes that have strong support for their involvement in bipolar disorder via gene expression. And there's several interesting genes here. For example, HTR6 on chromosome 1, MCHR1 on chromosome 22. These are known targets of antipsychotics. And reduced expression of furin is also associated with bipolar disorder, as it is in schizophrenia, where there's already been much functional genomics work done on the furin gene. Polygenic scores from the new GWAS results explain on average 4.75% of variance in European samples. And this is based on a series of leave one out polygenic scoring analyses across all of the data sets in our meta-analysis and looking at a weighted average across them. Looking at non-European data sets, in 2 East Asian samples, we saw about 2% variance explained, and in African American samples, 0.5% of variance. Here we're looking at some traits of interest which we selected for Mendelian randomization based on their relationship with bipolar disorder from clinical studies. So we were interested in sleep-related traits, smoking and alcohol use, educational attainment, and mood instability. And most of those show a pretty low genetic correlation with bipolar disorder. We use GSMR to do bidirectional Mendelian randomization to assess any evidence of causal relationships between these traits. So some of the most interesting results we saw is an association between bipolar disorder and longer sleep duration and reduced likelihood of being a morning person. Also, bipolar disorder and increased risk of problematic alcohol use and drinks per week, whereas the alcohol traits did not increase risk for bipolar disorder. We also saw a strong positive relationship between bipolar disorder and educational attainment, which is a well-observed epidemiological pattern. And we saw some evidence that cigarettes per day is associated with increased risk of bipolar disorder. And these results have been validated using other MR methods, and we've done several tests to ensure that there is no evidence of horizontal pleiotropy here. But despite this, we consider these more of an exploratory analysis really to investigate potential causal relationships with bipolar disorder that can be followed up with future experiments. So to summarize the current study, we have 42,000 cases in the most recent GWAS, 64 loci. If we look at the number of loci compared with the number of cases, you can see here that we have reached the inflection point now for bipolar disorder. Although having said that, we're still lagging behind schizophrenia in terms of the number of GWAS hits that we have, despite these two disorders having similar heritability and now having similar sample size in the GWAS. And this could be due to the heterogeneity that exists in bipolar disorder, with different subtypes having different genetic etiologies. And from the current study, we see several lines of evidence pointing towards the involvement of calcium channels, which could be an interesting avenue for future therapeutic development. And with that, I'd like to thank everyone in the bipolar disorder group, the PIs who contributed data, the analysts who have worked on the study, and Ole Andresen, who is chair of the bipolar disorder group.
Spectacular. Thank you so much, Niamh. We're, again, a little bit short on time, but maybe we have time for one or two questions. First one in the box is from Abe Palmer, who asked about whether lithium is represented in the drug classes that you saw enrichment for in your analysis.
Yeah, lithium is part of the psycholeptics, which was the most significantly enriched class. And I think of the subclasses, it's part of the antipsychotics. Those included mood stabilizers as well, which was the most enriched subclass of psycholeptics.